Hi class, in this recording we are going to be focusing on how neurons can be categorized based on their structure. In the previous video we talked about the structure of the neuron, the dendrites, the neurosoma, and the axons, and some variations within. After we're done talking about classifying neurons based on structure, we're then going to move on and focus on how neurons move things down their axon, which can be quite long. So let's dive in. As we're looking at the structural classification of neurons, we have a unipolar neuron. Uni is a prefix meaning one. When we think of pole, I want you to think of point or process. So a unipolar neuron is going to have one process coming off of it. And that one process coming off of the unipolar neuron typically is going to branch, and functionally we'll have an dendrite side and an axonal side. As we think of these unipolar neurons, they're typically going to be sensory cells associated with skin and or some organs in our body. They'll typically send information to the spinal cord. A lot of times, as we look at the unipolar neuron, there won't even be the dendrite side. We'll just have the neurosoma itself acting as the sensory receptor, and when stimulated, it sends a signal down the axonal side of its single pole to the central nervous system. We also can have an anaxonic neuron. These are the ones personally that I find to be rather confusing. Anaxonic neurons are going to be without an axon. This prefix "an" means without what? Without an axon. They have many dendrites, but no axon. Where do we find anaxic neurons? We find them primarily within the retina, the sensory receptor layer at the back of our eyeball that helps to turn photons of light into electrical signals. We have a bunch of anaxic neurons in our brain, and also within the adrenal glands directly above our kidneys. I personally am not very familiar with anaxic neurons. But if you have any questions, please let me know. I'll use that as an excuse to educate myself, and I'll get an answer to you to the best of my ability. We also can have multipolar neurons, and multipolar neurons are by far the most common. There are many variations of these multipolar neurons. For example, this one with a triangular shaped neurosoma is referred to as a pyramidal cell. And they, if we get to lab, you can see some pyramidal cells within the histology slides that we have in lab. Most of our multipolar neurons are going, particularly in a lab setting, are going to be on our ox spinal cord smears. And when we think of multipolar neurons, multi meaning more than two, they are going to have multiple extensions. So here's a great example. We can have lots and lots of dendrites, the one axon on our multipolar neurons. I'm going to emphasize that again. Multipolar neurons typically will have one axon and more than two dendrites. And just to review briefly, dendrites carry signals to the neurosoma. So here's the dendrites carrying signals to, drawing some arrows towards the neurosoma, and then the axon carries it away. There's an arrow going away from the neurosoma. We also could have a bipolar neuron, and as we look at these bipolar neurons are, well, going to have two extensions, bi meaning two. There's one axon and one dendrite. As we look at these bipolar neurons, we have a bunch of them in the in our mucous membrane of our nose. They are the olfactory cells. We also have bipolar neurons within our retina as well. We also have multipolar neurons within our retina. Our retinas are kind of complicated. And we also will have some bipolar neurons within the inner ear that help us with transmitting equilibrium and sound information from the inner ear to the central nervous system. Let's move on to axonal transport. This is the process of moving stuff from the neurosoma to the axon terminal. So we need to take those proteins, those substances that are manufactured in the neurosoma, and get them to the axon terminal where a lot of the action occurs. We also are going to have some issues with the axon. The axon can be up to 30 centimeters long. That's not uncommon to have a 30 centimeter long axon. And there's a lot of cell membrane associated with that. So we need to be able to repair that cell membrane. We also are going to need to maintain the gated ion channels within that axon to modulate or regulate electrical activity, and then take those neurotransmitters and enzymes made at the neurosoma and get them to the end, the axon terminal. So as we look at axonal transport, this is the ability to move stuff away from the neurosoma and towards the neurosoma. And it's a two-way process. We can have anterograde transport, where we take movement down the axon away from the neurosoma. So when we think of anterograde, I want you to think of away, A and A. Anterograde transport goes away from the neurosoma. We have retrograde transport as well. And when we think of retrograde transport, I want you to think of movement towards the axon, or excuse me, up the axon towards the neurosoma. So back towards the cell body of the neuron. And as we're moving stuff with the anterograde or retrograde motion through the axon, those substances are going to be guided by the microtubules of the cell membrane. And we have motor proteins, dynein and kinesin. Dynein is shown here, kinesin is shown down here. And these motor proteins will carry the transport vesicles on their back, so to speak. And then they'll just crawl or walk, almost like taking two fingers to make a little stick person and having it walk across the table. These proteins, dynein and kinesin, will walk along the microtubule. And because our microtubules have a directionality to them, they are not the same going forwards and backwards. Kinesin and dynein, these two proteins, will only travel in one direction. Kinesin will only go away from, think anterograde away. Kinesin only goes away from the neurosoma, and then dynein will go retrograde or back towards the neurosoma. And as we're looking at axonal transport, we can have fast axonal transport. This fast axonal transport is going to depend upon these motor proteins. So what are some of the things that will move? Well, let's talk about speeds. We can move between 20 and 400 millimeters per day. When we think of 400 millimeters, that's the same thing if we remove a zero as 40 centimeters per day. So relatively fast, considering that we have a microscopic motor taking nanometer steps down the length of this axon. I think that's pretty cool. And what kind of things do we move quickly? We are going to move organelles, enzymes, synaptic vesicles, and small molecules. And especially, I want to strongly emphasize the synaptic vesicles filled with a bunch of neurotransmitters, NT for neurotransmitters. And it's like I got cut off there. Let me try that again. NT for neurotransmitters. Synaptic vesicles filled with neurotransmitters makes up a very large amount of the anterograde transport. When we look at our retrograde transport back towards the neurosoma, we're going to be focusing on recycled materials. After neurotransmitters are released from the cell, they are destroyed in the axonal cleft, and then parts of them are reabsorbed and recycled and sent back to the neurosoma to be processed and turned back into a brand spanking new neurotransmitter. This makes up these recycled materials heading back towards the nucleus makes up a very large amount of the retrograde transport. However, pathogens will rely on this. If we think of rabies virus, herpes virus, tetanus, or polio viruses, all these different viruses are going to rely on retrograde transport to travel the length of the axon to get back to the neurosoma. So why is this significant? Because retrograde transport is going to take a while, and typically a couple days. It's not quite as fast as axonal transport. And as we're looking at this retrograde transport, this results in a delay between when we are infected and when the symptoms of that infection start to manifest themselves. We also can have slow axonal transport. And when we have this slow axonal transport, we're looking at up to 1 centimeter. So 10 millimeters, 1 centimeter per day, compared to 400 millimeters or 40 centimeters per day for the fast transport. So when we look at the slow transport, it's always going to be anterograde or away, A for away from the neurosoma. And we're going to be moving cytoskeletal components. The cytoskeletal components in particular, these are the things that the fast transport depends on. And it's kind of like building a road. Once you have a road in place, you can go across it quickly, but the act of building the road itself usually is pretty slow. We also have more miscellaneous enzymes, various enzymes being moved to the axon terminal. And we also can regenerate damaged axons as we are trying to send more axoplasm or cytoplasm over the axon down the axon. Now, remember, an axon is also known as a nerve fiber. So if we have a damaged axon, that's the same thing as saying that we have a damaged nerve fiber. And damaged nerve fibers or damaged axons regenerate at a speed that's governed by slow axonal transport. So up to a centimeter per day, but as little as 1/20th of a centimeter or half a millimeter per day is typically more common, particularly when we're thinking of regenerating a damaged nerve fiber. For any of you who've had surgery and you've had that numbness near the incision point, you know exactly what I'm talking about. Typically, it will take a couple weeks for your sensation to return near that incision site for your surgery. That's all we have for this recording. As always, if you have any questions, please feel free to post them on the class discussion board or shoot me an email, and happy studies.